Harvesting Energy, Glycolysis, and Cellular Respiration. This is Chapter 8. I am Colin McLeod. Raising a King. The skeleton of Richard III. He died around 500. Excuse me. I had something caught in my throat causing me to talk like Dick Van Dyke, who was an actor who did a terrible British accent imitation. Excuse me. Died around 500 years ago. It was rumored that he suffered horrific wounds in this battle, that he had a very extremely curved spine, and it is not exactly known where he was buried. So when these remains were unearthed, it was a little uncertain as to whether or not these remains really were those of King Richard III. He was ID'd using mitochondrial DNA found in the remains, and you might wonder, why was he ID'd using DNA in mitochondria? There's also DNA in the nucleus. There's much more DNA in the nucleus. That's where most of our DNA is. Well, think about this. In a single cell, there is just one nucleus, so one copy of all the chromosomes in there. But in a single cell, there can be many, many copies of mitochondrial DNA because there are many mitochondria in a single cell. For example, a single muscle cell may contain hundreds of mitochondria. So just one cell may have lots of mitochondrial DNA. So it is useful, it can be useful in identifying remains. We'll discuss why it's even more useful than that towards the end of this lecture. Quick review of how cells obtain energy and how all living things obtain energy. So let's remember that, well, number one, that solar energy, which is captured during photosynthesis, is the ultimate source of energy on Earth. Bullet 2, photosynthetic organisms capture the energy of sunlight and store it in the form of glucose. So the overall equation for photosynthesis is carbon dioxide and water produces glucose plus oxygen. Oxygen used to not exist in great abundance in the Earth's atmosphere when photosynthesis evolved. That is how oxygen began to accumulate in our atmosphere. It's about 18% of the air or the gas in our atmosphere today. Respiration uses glucose, C6H12O6, right here you can see, and gets the energy out of that. So a plant that produces glucose is actually producing that glucose for itself. Plants also have mitochondria, and they will respire that glucose to produce a large amount of ATP. That is chemical energy that can be used to do all kinds of cellular processes. The byproducts are carbon dioxide and water. Notice that these two equations are more or less the opposite of one another, which is why matter is recycled in ecosystems. Glucose. Glucose is a key energy storing molecule. Bullet number 2, nearly all cells, including ours, can metabolize glucose and extract energy from it. So plant cells, animal cells, the cells in the brain can in fact only metabolize glucose. So that is why we say I'm having a sugar crash. You're suffering from low blood sugar. Your thinking is a little bit foggy. That's because your brain is not getting enough glucose, and so it's not functioning at its optimum. So take-home message from this: you want to make sure that you're feeling like your blood glucose is right at about at a balanced level when you go into an exam. I remember when I was taking the SAT at the end of high school that I read up a bunch, or everybody's talking about, I guess a Snickers bar is just the right amount of sugar to keep you balanced out throughout that test. It's not like I talked a lot about it. I wasn't that big of a nerd. I was just like pretty much a huge nerd, but not that big a nerd. But anyway, you want to have some type of food source that will keep your blood sugar balanced for a little while. So like some peanuts and sugar might not be a bad idea for an exam, because all cells need glucose. Glucose metabolism is fairly simple. Last bullet, other organic molecules like amino acids or fats can also be metabolized and their energy extracted in ways similar to glucose. There are a few populations of Homo sapiens on Earth, such as those living in northern Alaska, Canada, and Russia, Siberia, those places, where people eat almost only meat, because that far north, not a lot of plants growing. Light, terrible. Temperature, terrible. So people who lived there back before technology made things different basically ate animals: seals, moose, bear, etc., fish, and they only had amino acids, and you can absolutely metabolize only proteins as well. Next, these structures here are all glucose. On the left, that is the ring form of glucose. In various conditions, it can change to being a linear sort of structure. I am not going to quiz you on the details of this. I just want you to have a little bit of a flexible ability to look at glucose in a couple different ways. Sometimes we'll also look at glucose in the last way on the right here as just a 6-carbon compound, because carbon is really the backbone of all organic compounds. So sometimes we're just really interested in how many carbons are there present. Overview of glucose breakdown. Number one, the main stages of glucose metabolism are glycolysis. This occurs in the cytosol of the cell, so no mitochondria involved in glycolysis. I'm going to say it again, and then you should repeat what I said. With glycolysis, the mitochondria is not involved. Say it to yourself. Okay. This process does not require oxygen. The respiration that occurs in the mitochondria does require oxygen. And we get a very low yield of ATP with glycolysis. So no mitochondria required and a low yield of ATP. Cellular respiration, this occurs inside the mitochondria of a cell. It requires oxygen and it produces a high yield of ATP. Remember endosymbiont theory? Mitochondria used to be free-living bacteria-like organisms that were able to use oxygen to break down things like glucose to produce a really high yield of ATP. They were like the powerhouse bacteria, and endosymbiont theory states or theorizes that long ago, a couple billion years ago, big cell phagocytized or tried to eat this little mitochondria, and instead of eating it, the mitochondria just started living inside that cell and produced a high yield of ATP, which that cell could use. The cell then in turn supplied the mitochondria with glucose, so it was a mutually beneficial relationship. Overview of glycolysis. So top here, circling it now, cytosol. This is where glycolysis happens. Just an overview here. We'll get into more details momentarily. We take one glucose molecule, and that's what we'll generally be talking about whenever we're talking about anything here is what happens to one glucose molecule. Glycolysis happens to it, and we get a low yield of ATP, 2 ATP. And if you're not sure why that's low, well, let's look in the mitochondria, the pinkish reddish thing here, circling it now. In the mitochondria, we'll get a total of 34 ATP. So glycolysis is 2 ATP. We definitely can call that a low yield of ATP for one molecule of glucose. Glucose goes through glycolysis. Glyco is a root word meaning sugar or sweet. Lysis means to split. So glucose is split into 2 molecules of pyruvate. And if no oxygen is present, such as maybe when you are sprinting and you're not breathing rapidly enough to supply all your muscle cells with oxygen, fermentation occurs in your muscles and produces this molecule up here, lactate, also called lactic acid, and that can contribute to your muscles being sore the next day, say if you are sprinting or if you are lifting an unusually heavy object repeatedly, i.e. weightlifting. If you are a yeast cell, firstly, congratulations on understanding English, but probably you can't understand this, so let's just talk about them as if they're not listening. Yeast cells will produce alcohol, ethanol. Ethanol is alcohol, same alcohol in beer and wine, and also carbon dioxide is a byproduct. That's why beer can be carbonated if you seal it in a kind of pressurized container, and the carbon dioxide will carbonate the beer. If oxygen is available, we move to cellular respiration, which goes on in the mitochondria, where we are going to use oxygen. I'm not going to require that you remember exactly how many oxygens are used or how many CO2s are produced. That's a little too much detail. So oxygen's used. We get some CO2, H2O as waste products. The CO2 is going to be excreted by breathing, and water is fine to just stay in the cell. Big bonus, so oh, big smiley face here, is these 34 ATP, which can be used to do all kinds of processes that are required inside of the cell. Details of glycolysis are a little bit more complex. So number one, there's an energy investment stage. So we've got one glucose molecule here, and we're going to invest 2 ATP. I think ATP is like money, yeah? 2 ATP, and we phosphorylate glucose 1 time, 2 times. So remember, these phosphates are like really high energy, and they increase the energetic state of glucose to a higher energetic state. So this is 1 fructose bisphosphate. And what else about that? Right. So glucose, we wouldn't want glucose just sort of spontaneously going through glycolysis or respiration. So glucose here is a little bit more stable, than fructose bisphosphate. So when we phosphorylate glucose, we increase its energy state and we lower the activation energy to split it in two. So after the energy investment stage, we're going to split it in two. So glucose molecule converted to highly reactive fructose bisphosphate. And if this sounds hard to remember, ose, that always means a carbohydrate, like glucose, carbs. Bis, that means 2. Phosphate means phosphate. So it's basically a carbohydrate with 2 phosphates on it. I will not require you to reconstruct all the root words here at all. Just kind of trying to help you break down that big word. So glucose molecule converted to highly reactive fructose bisphosphate by 2 enzyme catalyzed reactions. We can just say it's by an enzyme catalyzed reaction, and we use 2 ATPs. This lowers the activation energy for subsequent reactions. 2, the energy harvesting phase. So we've invested 2 ATP, gotten this molecule glucose so that it will undergo some reactions, and now we can get some energy out of it. So the energy harvesting phase is fructose bisphosphate is split, or lysis, right? Glycolysis, splitting a sugar, split into 2 3-carbon molecules of glyceraldehyde 3-phosphate. You can just think of that as G3P. That is totally good enough, G3P. And G3P is not particularly special. We didn't get any energy out of it, so we haven't harvested energy yet. But the next stage, let's direct our attention to this diagram down here. 2 G3Ps. The P stands for phosphate, right? So there's 1 phosphate on each G3P. They are going to be converted to pyruvate. They're going to be converted to pyruvate. And what happens during this phase is we are going to get some ATP produced. So one of these phosphates will come off of there, another phosphate will come off of there to add a third phosphate to this ADP, making ATP. That's nice. That can be used to power the cell. Secondly, let me rewind a little bit. You might have caught me up on a sort of error here. It's not an error, but it might look like one. There's only 2 phosphates here, so how is it possible to make 4 ATP? This is a dramatic simplification of glycolysis. There are many other intermediate substrates and products. So if that seems like it doesn't make sense, you're right, it doesn't make sense. So that's because we're simplifying it for you. So you can just think 4 ATP from glycolysis. Yay! Why? Well, because we get ATP. Good. Okay, nice. The more important thing, though, is this. In the energy harvesting phase, we are going to convert 2 NAD plus to 2 NADHs. What does that mean? Well, they are very short-term energy carriers. NAD plus is going to accept some hydrogens off of this G3P, and it's going to carry it to the electron transport chain to be discussed later. So in a series of reactions, as I just said, what I just said, same thing. Because 2 ATPs were used to activate the glucose molecule, there's only a net gain of 2 ATPs per glucose molecule, because in the energy investments phase, we used 2 ATP, and we produced 4, so it's a net gain of just 2 ATP per glucose molecule. Energy harvesting phase continued. So let me point out this molecule down here. If my video, my misshapen head is blocking part of it, it's not blocking the important part. The important part of glucose, pyruvate, is right here. The important part of pyruvate is right here. Why? Let's remember what we just said about NADH. NADH is going to carry high energy electrons and hydrogens to the electron transport chain. So pyruvate still has a lot more energy in it because we can see there's a hydrogen here, there's a second hydrogen here, and there's a third hydrogen there. So if oxygen is present, those hydrogens are why pyruvate is still considered sort of an energy dense molecule. It's still got more to give. So as each G3P is converted to pyruvate, two high energy electrons in a hydrogen ion are added to an empty electron carrier, NAD+, to make the high energy electron carrier molecule, NADH. Because two G3P molecules are produced per glucose molecule, two NADH carrier molecules are formed. And let me restate again, I'm not going to ask you to regurgitate the exact number of how many NADHs are produced or the exact number of how many ATPs are produced. Is it 4? It's the gross yield of 4 ATP, net yield of 2. This, I do need you to know this. It's a low yield, just meaning it's relatively low. It's not a lot. Little, few. So glycolysis in summary. The energy investment stage is the left part of this diagram. We use 2 ATPs to raise the energy of glucose by converting it to 1-fructose bisphosphate. This is a higher energy molecule, so it takes less energy for it to be split in the energy harvesting phase, glycolysis, the splitting of this sugar. When we split, after we've split 1-fructose bisphosphate into 2 G3Ps, we then are going to take some energy off of it to make ATP and NADH. Lastly, pyruvate. Pyruvate will go into the mitochondria if oxygen is present because it's still got a few more hydrogens that we can strip off of it using some more NADH and another molecule. Glycolysis in summary. If you're not tired of hearing about glycolysis yet, congratulations, we've got more. Yay. So the summary of it is pretty simple. Glucose is split into 2 pyruvates, right? So a 6-carbon sugar is split into 2 3-carbon sugars, right? See, there's 1, 2 of them, and each of them has 3 carbons. Sweet. We get 2 ATP net, and we get these high energy electron guys, NADH plus H plus. And if you're wondering what's up with that, it kind of carries it like an acid does. That's chemistry, and if it is frightening to think about all this chemistry and acid stuff, we can just ignore that. This is by 101. Each molecule is broken into 2 pyruvates. A net of 2 ATP and 2 NADHs are produced. The ATP can be used right away by the cell to do whatever it needs to do, and the energy will be used later on to help make a high yield of ATP in the electron transport chain inside of the mitochondria. Very similar to photosynthesis and what goes on there in the electron transport chain. Moving on to cellular respiration. If oxygen is present, these 2 pyruvate molecules are going to be broken down into 6 carbon dioxide molecules. We know that's one of the byproducts. And that's going to happen in the mitochondria and will produce a high yield of ATP, or 34 specifically. 3 locations we need to now be super solid on where they are. Number 1, the matrix. The matrix is the innermost part of the mitochondria. This little box there talks about it. It is a large volume, and that is suited to its function because there's going to be a lot of solutes and enzymes that all need to mix together to catalyze some reactions. The intermembrane space, I'll color some of that in. That's in this word box right here. But this is the intermembrane space, and it has a small volume, which is a good adaptation for its function because it is going to be where we're going to pump in H pluses. We're going to pump H pluses into there, similar to the way that they were pumped into the thylakoid space. It generates a high concentration of ions. That's going to be used for chemiosmosis. Then, of course, there's the outer membrane, which just provides a barrier to prevent those protons from leaving. 3, the inner membrane itself. The inner membrane itself, I'll trace that with a black line here. This is the inner membrane, and this is where the electron transport chain is. It's also where ATP synthase is. So essentially what's going to be happening is you're going to be pumping H pluses into the inner intermembrane space. Here we go. Cellular respiration, 2 stages. Number 1, we've still got this kind of high energy molecule pyruvate, and we're going to get all the rest of that energy off of there in 2 reactions. One is acetyl-CoA production. Acetyl coenzyme A is sometimes also used to describe this, and also the Krebs cycle. So these 2 things, acetyl-CoA production and Krebs cycle, they occur in the matrix. Matrix has a large volume, so all these reactions have got a lot of volume to get done in. They dissolve in water, right? One of the properties of water, good solvent. 2, the electron transport chain, and that's going to be along the inner membrane, right? This guy, this gal, it doesn't have a gender. It's just it's a thing. This thing here. That's where the electron transport chain occurs. Hey. So firstly, the getting more energy off of that pyruvate molecule after glycolysis. Pyruvate diffuses into the mitochondria and into the matrix space. So here's pyruvate. We've got those 3 carbons. There's a couple of hydrogens still attached to it somewhere, and that's what we're interested in getting because those are high energy bonds. Each pyruvate is split into 1 carbon dioxide and 1 acetyl-CoA, while generating 1 NADH. So the NADH generated here is what is useful. It's going to carry those protons and electrons or hydrogens to the electron transport chain. The acetyl-CoA here still has some more high energy electrons and hydrogens, so it is going to be processed further in the Krebs cycle, where we just continue doing the same thing. We're going to get more NADH. So here's the Krebs cycle, and again, this is a fantastic simplification. Let's not even talk about the real part of it because it's so many enzymes and substrates. It'll make your head spin. So the Krebs cycle. The acetyl-CoA enters a series of chemical reactions, still in the matrix, called the Krebs cycle. These reactions repeatedly remove high-energy electrons along with protons from organic molecules, transferring them to electron carriers NAD+ and also FAD. FAD, let's take a look at FAD. I'll circle it right here. You can see that. The FAD is going to gain 2 hydrogens. And I know that it's a lot to ask to get you to remember the structure of atoms. Remember that a hydrogen atom is 1 proton in the nucleus, and then it's got just 1 electron outside of here. This is 1 hydrogen. So sometimes we refer to them as a proton and a high energy electron. We can also refer to them as just a hydrogen, because a proton and an electron together make a hydrogen. Finally, 2 carbon dioxides and 1 ATP are generated per spin. So a little bit of ATP production, very little, just 1 ATP per spin. And again, I'm not going to ask you to memorize it's 1 here, it's 2 there, just very little, okay? I've included here the molecule pyruvate just to remind you that although pyruvate has been converted to, looking at the left-hand side here, acetyl-CoA, it has some of those high energy bonds, the hydrogen carbon bonds, still got some of those, maybe 1 less now because we lost 1 in the formation of acetyl-CoA. But that's what we're doing here. That's the bottom line. We're getting those hydrogen carbon bonds, the energy in them. So finally, we are going to take 3 NAD pluses and get 3 NADHs. So we've got some H's off of there, and that's with a couple acetyl-CoAs. Remember, don't need to add up all the numbers here. FAD is going to get some hydrogens as well. It's going to become FADH2. So those are some high energy hydrogens. Waste product, some carbon dioxide. This is all the carbon dioxide that glucose has to give. So all the carbons at this point have now been removed. Pyruvate, one pyruvate molecule. Remember, it's got 3 carbons. You can sort of see that in this diagram here. And 1 was removed in the formation of acetyl-CoA, and now the last 2 are removed in the Krebs cycle. So that makes a total of 3 carbons removed. Okay, so lastly, just a little bit of ATP produced. So the stage 1 reactions in summary, and why do we break it down into stages? Essentially because we want to make it clear what is really happening generally. And all these arrows and funny names, we're just doing this. We're getting NADH, and then also FADH2, FADH2, and these are just carrying energy temporarily. They're carrying these hydrogens very temporarily. So I'll circle where the NADHs are produced. NADH, 1 is produced per pyruvate molecule. I can't even remember the exact numbers. 1's produced per pyruvate molecule when we make coenzyme A. And then we get some more in the Krebs cycle. And we also get more NADH there. And then we also get some FADH2. Little bit of ATP produced as well. Could ask you, is ATP produced in the Krebs cycle? Yes, just very little. Okay, so now stage 2, the electron transport chain. I'll read through these just to summarize what we're going to be talking about and what we have talked about. At this point, most of the energy in glucose has been stored very temporarily in electron carriers, NADH and FADH2. Only 4 total ATP have actually been produced for glucose after complete breakdown in the Krebs cycle. So again, that's very little. It's not that much. Don't have to memorize all the numbers. Just very little, we're going to say. Only when we get our electron carriers, NADH and FADH2, to the electron transport chain can we use the high energy electrons they are carrying to make lots of ATP. So here it is, stage 2, the electron transport chain. I'll go through it slowly. That's a lot. You may want to pause this video as I talk about it and use the book to reinforce what I'm saying. So number 1, we're going to look right at the bottom here. Number 1, these high energy carriers, FADH and NADH2, they're going to donate their electrons to the electron transport chain. The electron transport chain is this gray arrow. I'll use a black thing to highlight it. This is the electron transport chain. This is where all those electrons are going to go until they eventually are accepted by oxygen. And that's why oxygen is required. Oxygen has to accept those electrons, or there's basically a traffic jam of electrons on that electron transport chain, now seen as a black arrow. Okay, what is the energy from these high energy electrons used to do? Okay, bottom left-hand corner, circling these 2 electrons there, and let me draw your attention to these hydrogens, or these protons, right? H+. There's another H+ here, and there's another H+ here, and another H+ here even. So the electrons from NADH and FADH2, that energy is going to be used to pump the H+ into the intermembrane space. That process is highlighted in red arrows, and I'll direct your attention to it by adding a black arrow. So that is where the energy from the electrons is being used to pump the H+ into the intermembrane space. That's here, Mr. Smiley Face. It's also written here, the intermembrane space. So what is going on here is we're getting a high concentration of protons in the intermembrane space, and that's everything above this membrane with all those blue blobs in there. That's everything above there. That's the intermembrane space. We get a high concentration of protons. What happens with them? We're going to use them because it's a kind of like potential energy, like water behind a dam, for chemiosmosis. And that is going to happen. I'll circle it, draw your attention to this. Chemiosmosis is going to happen in this kind of region below where it says chemiosmosis. And protons, the H pluses, will diffuse down through ATP synthase back into the matrix. And this diffusion of those H pluses drives the production of ATP using ADP and inorganic phosphate as substrates. ADP has 2 phosphates. Inorganic phosphate has 1. 1 plus 2 is 3, which makes adenosine triphosphate, ATP. I encourage you to pause the video, review in your book to reinforce what I have just stated. Why cyanide is so deadly. Cyanide, even in very small amounts, causes death. Why? Because all cells must produce a large amount of ATP or they die really rapidly. So here's cyanide. I've added it to the electron transport chain. Cyanide binds to one of the transport proteins that is moving these electrons along the transport chain. And when it binds, it basically causes an electron traffic jam. So all these electrons going along the electron transport chain follow the black line, right? They're going, and then they go, and then they all start honking their horns. What's the hold up here? And cyanide's just sitting in the road like a big rock, and the electrons can't keep going. So what that means is that no more electrons can be donated from NADH and FADH2. So again, all these electrons are just sitting there honking their horns. Hey, what's the hold up here? And this whole thing just shuts down. So no more pumping H plus into the inner membrane space. No, no, no. So no chemiosmosis as well, and no ATP production in the mitochondria. So cyanide just shuts down mitochondria, full stop, gridlock. Cool. At least I think so. So can you answer these questions about the electron transport chain? I would suggest pausing the video now, reading through them, using your book, or perhaps rewinding the lecture to help you answer these things. 1 through 4. Pause it. Try to answer them on your own. If you didn't pause it, it's too late now, because now I'm going to help you, and it'd be better if you worked through it. But if you want me to just tell you, okay, I'm a nice guy, I will. So what is the role of NADH and FADH2? These two molecules very generally carry high energy electrons as well as protons. So in the form of hydrogen, they carry energy temporarily from several different molecules, from glycolysis, those reactions, from the formation of acetyl-CoA, from that reaction, they carry high energy electrons from the Krebs cycle. They carry them to the electron transport chain in the mitochondria. Number 2, how's the H plus concentration gradient formed? The energy is from, well, glucose basically, right? But more specifically, it is from all those high energy electrons that were gradually stripped off of glucose and other molecules, pyruvate, the formation of coenzyme A, acetyl-CoA, stripped off of other things, various intermediate products. We get energy, and the energy is in the form of electrons. We use that energy to pump protons into the intermembrane space. 3, how is the H plus concentration gradient used? Synthesize ATP. Well, by chemiosmosis. Essentially, those H pluses diffuse from the intermembrane space into the matrix. That diffusion is used to power ATP synthase, which makes ATP. What's the role of oxygen? We can call it the terminal electron acceptor. It accepts the electrons at the end of the electron transport chain. And oxygen also, so it accepts the electrons off the electron transport chain. Those protons, or H pluses that diffused back into the matrix, those also bind to oxygen, and that's how we get the formation of H2O as the final byproduct. So summary of all the steps. Firstly, let me point out one thing generally. Glucose is what we start with. This is the food molecule, and it's converted to all these different things here throughout the Krebs cycle. I'll let you review the words, all those words in there yourself, but that's where all the food molecules are. What else is happening is we're stripping off high energy electrons along with their protons throughout glycolysis, the production of acetyl-CoA, and also through the Krebs cycle. All of them are transported to the, well, I got to find a different color here, the inner membrane where the electron transport chain is. So they're all transferred to the inner membrane where the electron transport chain is. Right there, and right there. That's what NADH and FADH2 do. Throughout the usage of glucose and all those other things, pyruvate, acetyl-CoA, we get a small amount of ATP in glycolysis, another small amount of ATP in the Krebs cycle, and then a lot of ATP from the electron transport chain and chemiosmosis, 32, or a lot. So, big smiley face here, okay? Big smiley face. Lots of ATP is produced there. Raising a king. Mitochondrial DNA has only 37 genes, which code for various parts of the electron transport chain, among other things. So that DNA in the mitochondria, it has a use, and not surprisingly, it's used to make stuff that the mitochondria need. And when the mitochondria were free-living, bacteria-like organisms long ago, that DNA was used to produce all the things the mitochondria need. So that's why they have that DNA in there, to make all those proteins and whatnot involved in the electron transport chain, ATP synthase, etc. The mitochondria cannot really survive outside of a cell. There's some evidence suggesting otherwise, but let's not get into that. And the reason for that is basically the nucleus of all cells supplies some of the stuff that a mitochondria needs. Mitochondria have just gradually gotten lazy over the billions of years they've been staying in this nice warm house called a eukaryotic cell. And the eukaryotic cell has evolved to help the mitochondria by producing various proteins and whatnot that the mitochondria need. So mitochondria can't really live by themselves. Sorry for that slip up into the Dick Van Dyke thing. I don't know what's up with that. What about other nutrients? So those people who lived in the far north of various parts of the world, they're only eating meat. Does that mean that they can't get any energy from it? Side note on that, there's not really much wrong with just eating meat. But here's the thing. Those people living in the north, they didn't just eat skeletal muscle like we do, right? Like what's in a hamburger, what's in steak. Some of us might eat various other parts of the animal, such as liver. I lived abroad for a while. I've eaten a whole load of different things. You name it, I will not spoil your dinner by telling you all the things I've eaten. Suffice to say this, the people living in the north, they ate everything in the animal, and all those other organs are tremendously nutritious. So if you eat liver, that's like eating a salad combined with a burger. It's really, really healthy for you. Another side note, most dog food is made from liver. Final side note is dog food more healthy than what we eat sometimes? Well, don't worry, that stuff's not going to be on the test, but I thought you might like to know that. Okay, so what about other nutrients? Number one, some amino acids can be converted directly into pyruvate. Remember that there are 20 amino acids, so it varies what happens to them. Suffice to say this, amino acids enter respiration at various stages. Some of them enter as pyruvate, some of them enter into the acetyl-CoA step, and then many of them go into the Krebs cycle. Fatty acids will generally just go, most of them, the fatty acids, right here, and there we are. The fatty acids will go into acetyl-CoA. That's where they are processed. Remember, fatty acids are long chains of carbons and hydrogens. I've shown you one right down here. That's a saturated fatty acid, and basically, Some enzymes will chop off 2 carbons along with their hydrogens at a time. So let's chop. Okay, chop. Chop complete. And we would make 1 acetyl-CoA. Then we chop the next one. Chop, chop, chop. And we get another acetyl-CoA. Then we chop this last one. Chop, chop, chop. And we get another acetyl-CoA. What happens with this last carbon? You don't really need to know, but if you're curious, it gets converted into carbon dioxide. Whee! Okay, glycerol, that is fed into glycolysis. Yay. So how can you get fat by eating sugar? Long story short, most chemical reactions are somewhat reversible. So if you're eating, say, lots of glucose, let's follow the arrows here. Glycolysis happens, pyruvate is produced, and pyruvate's converted to acetyl-CoA. I was just telling you how fatty acids can be converted to acetyl-CoA. Well, it turns out the reaction can be reversed. So if you're eating tons of glucose, so you're producing, let me erase some of these arrows now. So if you're eating lots of sugar, lots of glucose, then follow the arrows down, you're going to produce a lot of acetyl-CoA. And if your cell just doesn't need that much, then it's going to be, the acetyl-CoA is going to be converted to fatty acids, which are then converted to fat, which are stored in your fatty tissue, more accurately called adipose tissue. Yay, that's about that. So a little more detail on that. All of these reactions are reversible. So what about those fellas up in the north who were eating largely protein, which are composed of amino acids? And if they're eating animals, they're going to be eating a lot of fat too. I told you that your brain needs glucose or it will essentially die. Well, that's true. So fats could be converted, could enter into, let's see, either acetyl-CoA or glycolysis, and then the proteins, same thing. They could be converted to any one of these things: pyruvate, acetyl-CoA, Krebs cycle, and all these reactions can be reversed. So, going against traffic, we can just produce glucose for the brain. So that's why eating protein only, there's no problem supplying the body with blood sugar. All these reactions are reversible. Now, if you're looking at all these arrows and just hating me, no, I'm not going to ask you to remember all of these arrows. Not at all. What I do want you to know is this. Whatever food we're eating, it can basically be converted to any other food molecule, okay? All the enzymes are there. It's like a super awesome mechanic shop. They can take what you bring them and they can make something else out of it. That's the bottom line here. Okay. So why do we eat and breathe? All cellular processes and functions must have a ready supply of ATP, or they won't work right, or they won't work at all. That's why we eat, so that we can produce ATP. You need a constant supply of electrons to drive the ETC, electron transport chain. These come from the foods you eat, so electrons. So if somebody asks you why you're so hungry, you can just reply to them, electrons. You may look like a little bit of a pretentious meanie, but you can say that anyway. Next bullet, you must constantly breathe oxygen. You need this oxygen at the end of the electron transport chain in cellular respiration so that we can do chemiosmosis and get a large yield of ATP. If you stop breathing, or if a poison like cyanide stops your electron transport chain from working, you can't make much ATP and you die within minutes. Unlike other cells in your body, muscle cells can actually survive for a while, several minutes, hooray, without oxygen because they can switch and use fermentation when oxygen levels are low. That is why people who train for doing things like sprinting, their muscles actually get adapted to that sprinting type of energy usage. You can't breathe fast enough to supply your muscles with all the oxygen they're going to need for that 100-meter, 100-yard dash. You can't do it. In fact, you could probably run 100 meters holding your breath. How is that possible? Well, they just do fermentation or anaerobic respiration. No oxygen needed, and they store tons of glycogen. The muscles do. If somebody's really practicing to be a sprinter or a heavy weightlifter, they store glycogen, which can be converted to glucose really quick. So you get a lot of glucose, and you can just get a little bit of ATP off of each of those glucose molecules in glycolysis so that you can go, and then you're done, right? Okay, cool. Side note, my babble that is not that is not contained in these words, not on the exam, just thought you might like to know some extra stuff. So this stuff you do need to know. Anaerobic respiration, that means no O2, or fermentation. Same thing, different names. Annoying, isn't it? So number one, it is inefficient, just like glycolysis, but we get a rapid supply of ATP. Rapid ATP production doesn't have all those steps like the acetyl-CoA, Krebs cycle, electron transport chain. It's really fast. All organisms use glycolysis, all of them. It is very old, this biochemical pathway. Supports the theory of evolution, i.e. that if all organisms on Earth are essentially using the same enzymes, substrates, and products, suggests that we have a common ancestor, i.e. one cell. Pretty wild. But it is what the evidence suggests. So very old. And it suggests that all life evolved from a common ancestor. So taking a look at these pictures here, these are all examples where glycolysis will be the main supply of ATP. So as I mentioned, sprinting, these two ladies sprinting, that's going to be basically anaerobic respiration. Their muscle cells are just going to do glycolysis, rapid production of ATP, kind of inefficient, kind of a good way to lose weight too, because you kind of waste a lot of that energy in the muscles. Bacteria, absolutely, they can do anaerobic respiration. They can also do aerobic respiration. This fella running from the saber-toothed tiger, well, both would be happening here. He's certainly breathing hard, right? So some oxygen is being supplied to all the muscle cells. So there is some chemiosmosis, so a large supply of ATP, but the caveman or woman or whatever, I guess it's got to be a guy. The caveman can get a little extra amount of ATP production by doing some anaerobic respiration at the same time. That just gets a little bit more, and it might just save him from the saber-toothed tiger. So this is what's going to be happening when you have a great need of a little more energy for a short amount of time. Can't last long because a byproduct is lactic acid, which slowly poisons you. This is Saccharomyces cerevisiae. You don't need to remember that name. It's yeast, same yeast we use in making beer and wine, and it is almost only doing anaerobic respiration. They just tend to do that. Even when oxygen is present, they just tend to do that. Why? Not really sure, actually. Continuing, used by unicellular organisms before photosynthesis evolved, which filled the atmosphere with O2. So before chloroplasts started producing all that extra oxygen that they didn't use, this was the main way that cells would get ATP out of food molecules. This was the main way because oxygen wasn't available to accept those electrons. Many microorganisms still live in places such as our intestinal tract without oxygen. And those are typically considered healthy bacteria. Side note, quite an entertaining and rare disorder to have. Sometimes people will get an infection of the intestines of this yeast, the same yeast that is used to produce beer and wine, and you can predict the results. It's called autobrewery syndrome, meaning your intestines become like a small brewery. And people typically don't know that they have this disease or this infection. Their family members kind of start noticing that they behave sort of oddly. They may have extreme emotional swings. Generally, they're caught. The two instances I've read of it, they're caught because they're pulled over by the police. In both instances, they took a breathalyzer, and the police said, "You have about the equivalent of 10 servings of alcohol, and you're driving." To which the person responded, "I don't drink. You're crazy." Much yelling ensued. Eventually, they figured out that the person simply has a bizarre yeast infection that is producing alcohol in their intestines. Yikes. So how does fermentation allow glycolysis to continue when oxygen is lacking? Pyruvate is processed differently under anaerobic conditions. So pyruvate essentially becomes a waste product because it cannot be used in the production of acetyl-CoA at all. So it's essentially a waste product. When oxygen is lacking, pyruvate is going to be converted to either lactate or ethanol. Ethanol is the alcohol produced by yeast for beer and wine. And lactic acid is what makes our muscles feel sore. Lactate can also be called lactic acid. Both are correct. Fermentation does not produce more ATP, but it is necessary to regenerate the high energy electron carrier molecule NAD+, which must be available for glycolysis to continue. So when we were talking about glycolysis before, we were simply saying, well, there's NAD+, and it gets an H. So that's great, but remember, if NAD+ gets an H, it's not NAD+ anymore. It's kind of like when you fill up a pickup truck, the pickup truck's full now. It can't take anymore. Similar situation. Once NAD+ gets a hydrogen, it can't be used to do that again. Can't take more. Glycolysis is the common starting point for both respiration and fermentation. So, lactate fermentation. In animals and some microorganisms, pyruvate is converted to lactate or lactic acid, which regenerates a cell's supply of NAD+. So this whole process of producing lactic acid or doing anaerobic respiration, the whole process is about regenerating the cell supply of NAD+. No CO2 is produced. So what happens? The left-hand side of this diagram here, so where one glucose is used to make pyruvate, we get some ATP and NADH. That's just glycolysis that we've studied already. Now, it slowly uses up the cell supply of NAD+, so we need to get more somehow. So what happens is this. Instead of this NADH going to the electron transport chain, it's just sort of wasted instead. And what it does is NADH is going to donate its hydrogen, or high energy electron and proton, to pyruvate. And so pyruvate is just kind of wasted. It's converted to lactate, which is toxic. It's an acid, lactic acid. But the one good thing that happens here is NADH loses its H, producing NAD+ again. So we can regenerate the cell supply of NAD+ so that glycolysis can continue, and glycolysis does produce a small amount of ATP, which in some circumstances may allow the cell to survive, or the organism, such as the caveman, running away from the saber-toothed tiger, to just run a little bit faster for a little bit of time and survive. And here, what we're looking at is essentially the same thing I described in the last slide, except that I've shown on the bottom all of glycolysis. Glycolysis requires NAD+ to produce ATP. Quick review. Energy investment stage is when glycolysis needs a couple of ATP to lower the activation energy. We get 1-fructose bisphosphate. That is a higher energy molecule, more reactive. So it splits to form 2G3P. These 2G3P can be used to make some ATP. And that is helpful, can help the cell survive. A little more extra energy, maybe involved in muscle contraction or whatever. Energy harvesting stage, we also get 2 NADHs, and if we have oxygen present, this will go to the mitochondria. But if no oxygen is present, it just accumulates, and slowly NAD+ reserves are depleted. So on the top here, this is what was on the last slide. NADH, I'm going to circle it, bring your attention to this. NADH adds its hydrogen to pyruvate to produce a waste product that is toxic, lactic acid, but it's okay if it just lasts a short time. We thus get NAD+ back, regenerating our supply of NAD+. Glycolysis can continue. This is fermentation in yeast cells. Plant cells will also do this, and we get the production of ethanol. And by the way, no, there is no alcohol tree. No, it's not like there's an alcohol tree that just drips, drinkable alcohol. No. But plants will also do this. Yeast and plants will produce ethanol. So how does this happen? Basically, same story. Glucose in glycolysis is used to make pyruvate. We get ATP. The cell is happy. It can at least spend it to do some things that will help it survive. Pyruvate is now considered a waste product because no oxygen is present to enable the electron transport chain to work. Now in yeast cells, they convert pyruvate to ethanol. They convert it to ethanol and also carbon dioxide, which is why beer can be carbonated. The whole purpose of this is to use this NADH when oxygen isn't present to convert it back to NAD+. NAD+ is used in glycolysis, so glycolysis may continue. We're happy with glycolysis for now because we get a little bit of ATP produced. This is in yeast and plants, not animals. Animals do not produce ethanol. That would be really bizarre. That would mean that if you were sprinting and started doing anaerobic respiration, your muscles would produce alcohol and you would slowly become intoxicated. So obviously animals don't do this. Which of these are products of lactic acid fermentation and which of alcoholic fermentation? So here we go. This is sauerkraut. Sauerkraut, you may know, is a way to preserve cabbage and also make a tasty thing to eat. Sauerkraut. So it is sour because of an acid present in it. So it must be lactic acid fermentation. Some microorganisms other than yeast, so not yeast, in fact, bacteria, will do lactic acid fermentation. This produces lactic acid, and that lactic acid will prevent other microorganisms such as fungi and molds and nasty stuff like that from growing in sauerkraut. So that's why this is such an effective way to preserve cabbage. How about here? This is the production of cheese, and that again is going to be lactic acid fermentation. There isn't alcohol in cheese, and that is also another way to preserve dairy products, to preserve milk. Here, that is a couple of beers. That's obviously alcoholic fermentation. Wine, alcoholic fermentation. Yogurt, this would be lactic acid fermentation. And again, that lactic acid will help to preserve it, keep other microorganisms from growing in it. Pickles, again, lactic acid fermentation. Bacteria are making lactic acid, which prevents other organisms from growing in. That's one of the things that helps preserve pickles. Bread, this is actually alcoholic fermentation. That's why you add yeast to bread to help it rise. What causes it to rise is the fact that CO2 is produced within the bread, and as more gas is produced in that dough, the bread rises. Here, sore muscles. So this person's been doing some kind of shoulder lifting exercise, and the muscles are sore, and they are sore not because of the production of alcohol, but because of the production of lactic acid in their muscle cells. Raising a king. Isotopes occur in different ratios in different food. These isotopes become incorporated into tissues such as bones and teeth. Remember that an isotope is basically the same thing as it's an atom, and so like oxygen, for example, usually has a certain number of neutrons in the nucleus, and if it has a greater or lesser number of those neutrons, we call it an isotope. And they're generally much more rare than sort of the standard number of neutrons. Based on oxygen isotopes present in Richard III's bones, it appears in the last few years of his life, roughly 25% of the liquid he drank was wine produced by the alcoholic fermentation of grapes. Food fit for a king. Lastly, mitochondrial DNA only passed through the maternal line, so from the mom only. Typically, we think of our DNA as half from mom, half from dad, which is true, with the exception of mitochondrial DNA, because the sperm's mitochondria do not enter the egg. The sperm gets to the egg and only inserts its nucleus into the egg cell. So no other organelles are inserted, including the mitochondria. Turns out that there is one known descendant from Richard III's mom who's still alive today. And it turns out that his mitochondrial DNA is 100% the same as the mitochondrial DNA thought to be Richard III's in that skeleton that was found underneath a parking lot. The DNA evidence, along with the fact that the location, skeletal deformities such as the very curved spine, and also battle wounds provide very convincing evidence that the skeleton is indeed Richard III's. And that concludes our lecture on respiration. Hope you enjoyed it. Have a nice day.